we sorted some things out. Welcome to another session of our Evolution Seminar Series. We're very excited to have Audrey Gasch today, who's a professor of medical genetics and director of the Center for Genomic Science Innovation here at UW-Madison. He's going to be talking to us about yeast chromosome duplication. So without further ado, let's jump right into the science. Please join me in welcoming Professor Audrey Gasch.
Great, thanks. And are we good with the microphone and everybody online can hear? Great. Great. Well, thank you, Yasim and Emily and the whole organizing committee for inviting me to speak. This is kind of a fun situation and a weird situation because I'm giving the talk here, but actually people in my lab are the ones who did all of the work. And so I know we have James at least in the back, and you can see him here on the far left of this partial image. Julie Rojas is a postdoc. I'm going to talk a lot about their work today. Now, I know that this is also tied to a class. How many of you guys are in the class that we're going to meet afterwards? So a lot of the students here. Great. Well, hopefully this is makes sense to you guys. I was asked to give, you guys are all, I'm told, excellent researchers. And so I was asked to give just a regular research talk. I'm going to give a talk that I've given recently to another institution. But this is casual. So if you guys have questions, if anybody has questions, feel free just to raise your hand and shout them out. Okay. Now, let's see. So I'm not sure, can I advance? Yeah, I guess so. So in the Gasch lab, we are really interested in addressing fundamental questions in genotype, phenotype, environment interaction. So we're interested in how different organisms evolve different forms and functions, and how their interaction with the environment affects that evolution. Because of course, we don't live in a vacuum. All of us are living in environments that are dynamic and can vary in space and time. And our lab has been particularly interested in stressful environmental situations because it turns out often there are stresses in the environment and organisms have to be able to deal with that. So we ask a lot of questions about, we primarily use budding yeast, Saccharomyces cerevisiae, and we're interested in understanding really basic things about how cells manage environmental stress and how they sense it. How do cells know when there's a problem? They have to be able to integrate many different features from their internal and external environment and then mount the correct response. That's often a systems-wide response. So cells will change their transcription, their translation, protein modifications, the functions of those proteins. These things can together mediate what I like to think of as higher level functions, metabolic fluxes, for example, cell division properties, growth rate, etc. So it turns out it's still not really understood how cells do this on a systems-wide level. So we spent a lot of time thinking about that. But what I'm going to focus on today is actually something we kind of fell into a few years ago, and that is a different type of stress. And that is the stress, an internal stressor, the stress of aneuploidy. So aneuploidy is a situation where cells have an abnormal number of one or more chromosomes. Many people have heard of aneuploidy in the form of Down syndrome. And this is where people have an abnormal number of, or an extra copy of chromosome 21. Let's see if I can get let me get a pointer here. And so this is a human karyotype. You see the chromosomes have been colorized. There's an extra copy of chromosome 21. And what we know is that during mammalian development, having an extra copy of one chromosome is actually really deleterious to the person who's carrying this, right? Medicine has done a lot to improve the lives of people with Down syndrome, but they still have a lot of medical complications. So the flip side is that we know that many cancer cells have abnormal karyotypes. They often amplify chromosomes all over the place. And although that's bad for the person who's got cancer, the tumor actually benefits from having an abnormal number of chromosomes. You see a melanoma karyotype is just crazy amplifications of a bunch of chromosomes. So why is that? We now have definitive proof that many tumor cells actually get a benefit from duplicating their chromosomes. They're often more deleterious to the person, but those tumors can pick up drug resistance. And this is really important for thinking about evolution, because it turns out also in nature there are cases, it's certainly for pathogenic fungi, this is really, really clear, where cells can pick up extra chromosomes and very rapidly get benefits of that. So drug tolerance is a really clear example. We have fungal pathogens that infect people that are drug resistant to the drug treatments, and a leading cause of drug resistance is those drug resistant fungi have duplicated chromosomes. So a lot of people in the aneuploidy field have described this as a dichotomy. How can aneuploidy be bad in one situation and good in another situation? Well, there are several different ways to explain that. But in the context of evolution, it may be actually not, maybe it's not a dichotomy, it makes perfect sense. There can be a cost to having extra chromosomes. And when I say aneuploidy, for the rest of the talk, I'm going to focus on when cells have extra of one or more chromosomes. So there can, in perfect conditions, there's a cost to that. But if the environment changes, for example, suddenly there's a drug that the microbe has to be able to respond to, suddenly now the selective pressures change and the cells can actually have a benefit from that. And so we are really interested in understanding the cost-benefit balance in aneuploidy, and we're using budding yeast, Saccharomyces cerevisiae, as a model to understand that. The challenge is, after all this time, whether we're talking about people or yeast, we as a field still don't really understand the consequences, the cellular consequences of having abnormal number of chromosomes. Why is there a cost to that? Why is it deleterious? When is it beneficial? We need to understand the molecular details to really get that. So there's been seminal work done by other labs in yeast, using yeast as a model for eukaryotic cellular biology. And what many of those studies, I'll come back to this in a minute, have studied laboratory strains of yeast, one in particular called W303. And what the field has seen is that when this organism, this particular lab strain of yeast has extra chromosomes, it's very, very bad. Regardless of which chromosome is duplicated. Cells have a severe growth disadvantage, all kinds of signatures of the stress response, even though the stress now is coming from inside the cell. Lots of other issues, but what many people have focused on are these that I put in the boxes here, and that is proteostasis stress. So proteostasis refers to the proper balance of protein production, turnover, and management. And so one hypothesis in the field is that when cells have extra chromosomes, they're making too many proteins that are encoded on those chromosomes, and then the cells can't manage having extra proteins. That leads to all kinds of problems managing the protein proteostasis situation, and you get protein aggregates and toxicity. Although I will add, that model is not fully tested. It's just what people think makes sense. What we do know is that this laboratory strain, if you duplicate chromosomes, they have all kinds of protein stress. The problem, or the issue that we kind of, again, fell into a few years ago, is that everybody who has been studying aneuploidy yeast was studying laboratory strains of yeast that have been essentially what we call domesticated. They've just been grown in the lab for so long, they kind of adapted to the lab setting. So we have been studying wild strains of Saccharomyces cerevisiae to understand more about genotype-phenotype evolution. And in the process, we realized that a decent number of wild strains have extra chromosomes, but they seem to grow pretty fine. Nothing like the laboratory strain. And so I'm just going to not go into a lot of details, but I'll mention that we discovered the reason why the lab strain looks so different from wild strains. A lot of this was work that was led by James. And what we found was that the lab strain is an outlier. It's missing a critical gene called SSD1. And SSD1, that gene encodes a protein that is an RNA binding protein. So this protein binds to specific RNAs, it binds a specific sequence. We still don't know exactly what SSD1 is doing. We were not expecting to get an RNA binding protein out of this. This protein is, nobody knows exactly what it's doing, but it's been implicated in translational regulation. And that's maybe important for later on. And so the bottom line here is if we take wild strains and we delete this single gene, suddenly we can recapitulate essentially all of the phenotypes we looked at that had been reported to be a general aneuploidy signature in the lab strain. Now we know the lab strain is screwed up. Does that mean all of this knowledge that we've generated is wrong or bad? No, it's just specific for this strain. We've learned a lot about how a super aneuploidy sensitive strain has responded to stress. It hasn't really told us about normal strains. So now we come back to this question. Now that we have wild strains that have extra chromosomes, we think they're more representative of eukaryotes. What is the problem? Is there a cost to having extra chromosomes? And if so, why is there that cost? So yeast is a great model to study because we have all, as several people know who work on yeast, because we can very sensitively modulate the genome of cells, we can very sensitively phenotype them, and we can ask cool questions. And we can also come up with new hypotheses and then test those questions. And I'm going to lay that out for you guys today. So is there a cost to having extra chromosomes? There certainly were implications in the literature about that. I'm not going to go over all of these slides, but people had realized that cells that have larger chromosomes seem to have a bigger defect in their growth rate, but nobody really had a systematic way of studying that before. And so here's one of the first examples of the power of yeast. This yeast, Saccharomyces cerevisiae, has 16 different chromosomes, and it turns out we can manipulate their genome and engineer cells to duplicate any chromosome that we want. And so James and Julie, who I introduced at the beginning, some of you came in late, but maybe I'll restate because I think Julie was one of the people who came in late. This is a weird situation for me to give this talk because Julie and James, and I'm going to talk about Eduardo's work later too, did all of the work that went into this. So you guys should correct me if I say anything that's wrong. But so what we did is initiated by James. We used a method from the literature. A guy named Carey Bloom realized that if you drop a high copy promoter right next to the centromere, so for you undergrads in the lab, you maybe have studied chromosome biology. There's a centromeric region. You attach microtubules to that centromere, and then the chromosomes get pulled apart during division. And then you have two cells that are supposed to both have a balanced genome. It turns out in yeast, the centromeres are very short. If you put one of those promoters, a galactose-inducible promoter next to the centromere, and you induce a lot of transcription over the centromere, even just for one division, it screws up the attachment of the microtubules, and then the cell doesn't segregate the chromosome correctly. So with a lot of sensitive, super careful monitoring and characterization that James has really overseen in the lab, we were able to make a panel of yeast cells. So everything I'm going to talk about today, I think everything I'm going to talk about in the first part of my talk was done in a strain of S. cerevisiae that was isolated from oak tree soil. So it's a particular wild strain. And so what these guys did then was not only duplicated, so again, we have a panel of strains, a bunch of individual strains that are all isogenic, meaning their genome should be the same, but they've duplicated each chromosome one at a time. And James and Julie measured very sensitively the growth rate of these different strains and compared each of the aneuploids to the euploid with a normal karyotype. And so here what we're showing is the relative growth. You see the euploid grows at 100% on our scale. And then for all of these different aneuploids, their growth rate is reduced to different levels. Some chromosomes, when it's duplicated, the cells grow a lot slower. So we say there's a higher cost to that duplication. You'll notice this blank spot here. Chromosome 6 is so toxic that we weren't even able to make the duplication. And that's consistent with what other people have seen in the literature. We think it's because there are a couple of genes on that chromosome that are super toxic. Okay, anyway, so now the point is here, we've measured the fitness cost of duplicating each chromosome. These guys also very nicely did the same thing in the same genetic background, except that the pink bars represent the genetic background that's missing this RNA binding protein. Okay, so now you see that some of the chromosomes, so chromosome duplications, really need SSD1, like chromosome 4. If you don't have SSD1, the growth rate is way slower. And here we see that for chromosome 16, you can't even make that duplication in a strain that doesn't have this RNA binding protein. So that's great. So now we've got all these measured fitness costs. Remember, I said we don't really, so that's great. What's the cost of chromosome duplication? In this particular strain and these particular media conditions, super rich media, the optimal conditions for yeast, this is the cost if we measure cost in growth rate. But now we want to know why. Why are some chromosomes more deleterious than others when duplicated? Well, it's useful when you're doing research to think about the possibilities beforehand. And so we've thought a lot about different hypotheses. What are the models for why chromosome duplication might be bad? Well, on the one end of the spectrum is what we call the dosage sensitive gene model. So here is a really nice diagram that Julie made showing the euploid cell on the top, 2 different chromosomes, a gray chromosome and a yellow chromosome, and the representation of the aneuploid where we duplicated the yellow chromosome. So this model is that maybe there are just a few genes that are really toxic when duplicated. So if you duplicate the whole chromosome, you've duplicated that 1 or 2 really deleterious genes, and then that's all you need to get a sensitive cell. The other end of the spectrum, though, is what we call the genic load model. The chromosomes that have, but maybe all genes are toxic because they have some cost to their production, making them, transcribing them, translating them, etc. And the chromosomes that just have more genes have a bigger load for the cell, and they're going to be more toxic. So that's kind of the opposite end of the spectrum. It's really all the genes. Now, regardless of which mechanism we're talking about, one of the big theories in the literature is that it's called the balance hypothesis. And that is that the cell is optimized, or at least it's evolved to the state that it is. And it really is optimized for a balanced amount of all of the proteins in the cell, or at least some of the proteins. So for example, what people really focus on with the balance hypothesis is maybe there are some protein complexes. The ribosome is a perfect example. There are many different subunits in the ribosome. And we know that the way the ribosome is assembled is very precise order and relies on the precise amount of each of the 140 proteins, whatever it is, that are in that complex. So the balance hypothesis suggests that if you have an imbalance, it's not really about necessarily the total protein amounts, it's about the imbalance of certain subunits with regard to the rest. Like maybe that's what is really toxic. But anyway, the balance hypothesis is a big deal in terms of a model for why aneuploidy might be deleterious. Any questions at this point? Everybody following so far? Okay, great. So here is where Julie did lots of fancy footwork. So what her project has been doing, remember we measured the fitness costs already for chromosome duplication in this strain background. Now she's going to use mathematical modeling to try to predict the fitness costs and see how well that matches the measured fitness costs. Why are we doing that? Because the model that goes into the math is going to tell us how correct we were in guessing at what are the features that are important for the aneuploidy fitness. So hopefully that makes sense as we go through it. So the first thing that Julie did, it's really based on the hypothesis that I told you before. Maybe bigger chromosomes with more genes are going to be more toxic. So in the diagrams that I'm going to show you, remember I showed the relative growth rate in the bar graph before. Here, that's the y-axis. And so in this figure, the x-axis is just the number of genes on each chromosome. And then blue points were the panel that is the wild type background that has SSD1, and pink is the same strain, but it's missing SSD1. And then she simply did a linear fit. And you can see, hopefully, that these points actually fit a line pretty well. Chromosome 4 is the biggest chromosome and has the most genes, and it is among the more toxic. Lower point on the y-axis means there's a bigger fitness cost because there's a slower growth rate, okay? For those of you who have studied statistics before, you might be familiar with simple linear modeling. We can actually say, how well does this line explain the bouncing around of the blue points and the pink points? And you can get an R² value. And the way that we interpret that is that that value tells you how much of the differences in the growth rates can be explained by this linear model. And so in the wild type data, 60%, 62% of the variance can be explained with this model. The SSD1 mutant is more sensitive. You see that the line is steeper, and actually we can explain a bigger amount of variance, partly influenced by that steeper slope. So right away, this tells us that this super simple model, just counting the number of genes, actually can kind of explain the data, but not fully. So then the next thing that Julie wanted to know is, well, what about the actual genes that are on each chromosome? Might it matter that some genes are just a lot more toxic than others? And so here's where we use a really cool approach. And I should say that this data was generated by a graduate student in the lab, August Dutcher, who was nice enough to share the data. She's using this data for other purposes as well. But what we can do in yeast is we can take, we have a library of plasmids in which each gene from yeast has been cloned on the plasmid. So we can now put that plasmid, not just one plasmid, but we have one for every gene in yeast, let's say. We can put those plasmids into our oak soil strain that has a normal baseline karyotype. And what we can do is, maybe I'll go over this quickly. If there are questions in the class afterward, we can go through it. The plasmids also have what we call a DNA barcode. It's a synthetic sequence not found in the yeast genome, but it means that we experimenters can use that DNA sequence. There's a unique sequence for every single plasmid in the library. So that means we can put the plasmid library into the strain. Each cell takes up one plasmid. So now what it means is that each cell has duplicated only one gene at a time. Each cell is different. We can tell which gene because we can sequence the barcodes. So for now, all you need to know is this little colorized cartoon here means that each yeast of our oak soil strain has got duplicated a different gene. And now we grow that culture over a long time, many generations. Well, 10 generations, not that many. But now any gene that's been duplicated that's deleterious to the cells causes the cells to grow slower. Whereas other cells in the pool will grow perfectly fine and they'll outcompete those cells carrying deleterious genes. So the bottom line is, if you didn't understand any of that, all you need to know is we now can measure the cost of duplicating each gene one at a time. So now we come back to our aneuploidy case. I'll skip over this for now. So Julie now tried a new model. Remember, we have the measured fitness costs. We want to see if we can better use modeling to predict what those costs would be. But now instead of just summing up the number of genes per chromosome, she's summing up the fitness costs that we measured for duplicating each of those genes one at a time. And so now what she finds, the plot looks probably the same. You get some fits, but the important point is down here. And that is that for the wild type cells, the first model where we just count up the genes explains 62% of the variance. This model, where we consider what the genes are, at least in terms of their fitness costs, explains 69% of the variance. I won't go into the statistics, but this turns out to be a statistically significant improvement in the model. And actually, if you look at the SSD mutant, you see a similar thing. So what this means, the punchline is that when we consider the fitness costs of the individual genes, that better explains the measured fitness cost. So this already tells us something. The actual genes on the chromosomes matter. Any questions about that? Does that make sense? Okay. And so maybe for the sake of time, I want to be sure there's plenty of time for discussion. I will just throw it up there. Is the model significantly better? Julie's tested that in a couple of different ways, including random permutations, and you can just believe me that the model is better. Any questions at this point? Everybody gets the idea. So this already is telling us something, that the different genes have different impact on fitness when they're duplicated. But so now chromosomes have other stuff besides just genes. So what about the non-genic features? Other types of RNAs, origins of replication, these transposable elements. And so what Julie did was used a particular method called lasso regression. It is a way where you can essentially make a linear model with multiple different features included in the model, but the lasso regression actually chooses which are the important features to go in the model. Is that good enough, Julie? So for those of you who are new to all of this stuff, you probably have learned linear regression. I just showed that, where you would just have one feature and you're trying to fit the data to a line. It's harder to conceptualize, but you can make models like that that have multiple different things that all help to explain the data. So again, Julie used lasso regression. I'll skip over a lot of the details. She had a bunch of different features, and the lasso tells her that only a handful of those features are important. And so this diagram here is showing which features the model says are important in terms of explaining the aneuploidy cost. Those that are deleterious are skewed to the left. Those features that are beneficial are skewed to the right. And you get similar things if you're looking at the wild type strain background or the SSD minus background. So what do we find? First thing we find is that the most impactful feature in terms of explaining the real fitness costs is the additive gene fitness contributions, what I just showed you from the previous model. But now what the model tells us is actually SNO RNAs, small nuclear RNAs, the particular type of non-coding RNA that I'll introduce in a little more detail in a minute, one class of non-coding RNA, the model says that those are deleterious. Chromosomes that have more SNO RNAs are more deleterious than you would predict if you only consider the genes. Makes sense? Now, meanwhile, the model actually told us something surprising, and that is that the model is telling us that there. Is that good now? And so the model, is everything working now? Okay, great. The model tells us that in addition to the genes, SNO RNAs are predicted to be deleterious, but tRNAs are actually predicted to be beneficial. What that means is that the model is telling us that chromosomes that have more tRNAs on them, so duplicating those tRNAs, actually are doing better than the model would otherwise predict. So that suggests, the model is suggesting that more tRNAs is good in the aneuploidy setting. All right, so I think I need my laser pointer. And so now this is just to show, again, more linear plots. But what's important down here are the R² values. So now Julie has model 3, which has added the genes, the contribution of the SNO RNAs, the contribution of the tRNAs, and now she can explain for the wild type 74% of the variation in the growth rates, and in the mutant 93% of the differences in the growth rate. So still not 100% explained. We would never expect a model to do 100%, but it's kind of amazing actually. We're starting to build up a model that is, we hope, telling us what's important. Now, the problem with models is that, how do you know the model's right? And so in a lot of modeling stuff, many of you may be aware, big data and modeling and AI is very, very trendy right now. How do you know those predictions are right? Often you don't. But in yeast, the amazing thing is we can go in and test some of these hypotheses, and that's what Julie has done. The first thing she wanted to test is it really true that duplicating the SNO RNAs is deleterious? So here, let me introduce the SNO RNAs here. So there are two different types of SNO RNAs. One is called the CD box SNO RNAs. These are RNAs that are incorporated into larger protein RNA complexes, and they actually help direct the complex towards specific RNAs that can be chemically modified. So CD box RNAs can direct this complex to RNAs that get methylated, and the HACA box SNO RNAs direct a different complex towards RNAs that get a pseudouridylation, different chemical molecule. And so whatever, these modifications help these RNAs get regulated appropriately. So the first question, is it true that duplicating some of the SNO RNAs is bad? So what Julie did is she cloned two different cassettes of SNO RNAs. Conveniently, chromosome 13 has a bunch of the CD box SNO RNAs in a row. So Julie just cloned those onto a plasmid. Chromosome 15 is a group of 7 of the other type of SNO RNAs. She cloned those. And what happens if you put these into yeast, these two different cassettes? Well, first thing that she found, here we have our growth rate. She's putting this into euploid cells that aren't aneuploid. They have a normal karyotype. And lo and behold, if you put either of these cassettes into cells, they grow slower. And that's statistically significant. So right here, this is telling us duplicating a subset of the SNO RNAs, at least these SNO RNAs, is deleterious. And in fact, if she goes on to do that, put the plasmids into the aneuploid cells, what we see is that in most of the cases, it's also deleterious. So this, we would say, check box. This is, at least to this extent, has validated the model that at least for these SNO RNAs, if you duplicate them, it's deleterious. Oh, actually, and then she did the reciprocal. This is a great thing about science. Anytime you can do kind of reciprocal experiments to show that you can get that answer in both directions is good. So here what Julie did was she took the cells, either the wild type cells or the SSD minus cells that have the extra copy of chromosome 13. You remember this has one of those cassettes on it. And what Julie did is, you have 2 copies of chromosome 13. She just deleted one of the copies of that SNO RNA. Everything else is still aneuploid. Now, in the wild type, it's a pretty subtle effect, but at least in the SSD minus strain, where they're more sensitive to chromosome 13 duplication, if you delete just the SNO RNAs, their growth rate is significantly improved. So you see how we're coming at this question from 2 different angles. And so that's even better, really suggests that at least, again, chromosome 13 SNO RNAs are detrimental. So now what about the tRNAs? You remember that the model is telling us that chromosomes with more tRNAs actually do better than expected. Now we can talk about the whys later. We're still trying to figure out the why. Right now we're trying to test, is this prediction true? That the more tRNAs is beneficial. So first thing Julie did was she got a plasmid from another lab, the Chi lab, where they had cloned all of the tRNAs from chromosome 12 on one giant plasmid. And so why did we choose the chromosome 12 RNAs? Because we had that, that's what we had. That's what we had on the plasmid. So if you put that, so here I'm showing the growth rates, Julie is showing her slide in a little bit of a different way. It's the growth rate of the cells that have the tRNA plasmid normalized to the same strain with the empty vector. So what we expect, here's the 100%. If these cells with the tRNAs are benefiting, we would expect the bar to be over 100%. But in fact, we don't see that. We see that the euploid cells with the tRNAs actually growing slower than the empty vector. So the euploid does not like the extra tRNAs. What about the aneuploids? Well, here's actually where it gets a little weird. In blue are the aneuploid growth rates for the wild type strain background. If we put the chromosome 12 tRNAs in that strain, they grow worse. That's these down arrows. However, they grow less worse, at least for chromosome aneuploids, that is chromosome 13 and 15, they grow less worse than the euploid. What does that mean? Not entirely clear. But now if you look at the pink bars, the SSD minus cells with the extra chromosome 12, actually the chromosome 4 with, again, in the SSD minus background, with the tRNAs, going a little bit slower, but it's way not the same as the wild type strain. But here we see for chromosome 13 and chromosome 15 aneuploids, the little green arrow here means that they actually are growing a little bit better if you put more tRNAs in there. So what's the conclusion with the model? Kind of mixed results at this point. And it's all specific to chromosome 12 tRNAs. But then what Julie did, we got advice from a colleague who works on tRNAs, and she, Anita Hopper, and she said, oh, well, you could try deleting this gene MAF1, which is a repressor of all of the tRNAs. So if you delete MAF1, it's supposed to upregulate all of the tRNAs in the cell. And so it turns out, once again, we're showing the relative growth rate here, and the wild type in blue and the SSD minus strain. And once again, the euploid, there's no effect. But for both the chromosome 13 and chromosome 15 aneuploids, the effect is clearest in the SSD minus strain. You see it's going over the 100%. So the SSD minus mutant, if it has these extra chromosomes, actually does better if you de-repress all of the tRNAs. Wild type, not a big signal. So now we have kind of mixed results with the tRNAs. The model predicted that they are beneficial in both the wild type strain background and the SSD minus strain, but we're really only seeing a positive effect in the SSD minus background. Why might that be? There are several possibilities. One is that maybe the model is wrong about the wild type and the extra tRNAs really only benefits the SSD minus strain. The model was still chosen in the wild type. Another possibility is just we have more room to detect an improvement in the SSD mutant because it's so much more sick if we duplicate chromosomes. And I think that's what I'm favoring at this point, but it's a little hard to distinguish. Okay. So now, oh, I thought I had animation, but that's all right. In the last, oh, I guess I got a little bit. Do we go till 1:30 officially? Yeah. Okay. So I want to leave time for discussion, but I want to talk about the end of Julie's project and then some bigger implications. So now we have this model for why aneuploidy is deleterious. We're still trying to figure out the why are tRNAs better, might be helping cells, etc. But now what we're trying to do is actually get a model to predict aneuploidy toxicity where we don't need the measured gene fitness costs. We have a purely computational model. So to do that, what we're trying to do, you will remember the model told us one of the most important things is the gene contributions to fitness. So what Julie is trying to do is develop a model to tell us can we predict what genes are going to be toxic when duplicated. So we're still kind of in the middle of this, but I think one of maybe the punchline there, I can answer questions about it later on. The punchline is that there isn't a strong, for those of you who are familiar with bioinformatics, what we can often do is say, well, we know that these genes are deleterious when duplicated because we've measured them. Now, can we use machine learning models to predict what are the features of those genes that distinguishes them from the neutral genes? Does that make sense? So often we do things like gene ontology enrichment. Has anybody heard of Go enrichment before? Maybe some people are not. So you can say, for the group that's deleterious, are there any functions that are overrepresented in that group? Like gene functions like ribosomal proteins or proteasome proteins, whatever. We're not really seeing any strong signature of that. So what does that tell us? If we believe that modeling, it might mean that you can't really predict just from the known function of the protein, which ones are going to be deleterious. Now what we're looking at is trying to find biophysical properties of those proteins. That kind of makes more sense. Maybe certain types of proteins, this was already in the literature, proteins with intrinsically disordered regions. You may know that many proteins fold up into a defined fold. And we can use a crystal structure to look at that fold, or now AlphaFold can predict it for you. But there are a lot of regions that aren't in a rigid structure. They're like flopping around. And that flopping around is probably important for their biology, but also it might actually make them more toxic when overexpressed. Several groups have found that, and Julie sees a weak signature there. But nothing really telling us like, this is what will make a protein toxic when it's overexpressed. But the strongest predictor is actually deleterious genes tend to be longer. And so what Julie can, do now is she's looking at a bunch of different features. Down here, you see gene length. This is the group of genes that are deleterious are statistically significantly longer than the ones that aren't. That doesn't mean every single gene is longer, but as a group, they tend to be longer. There's a very strong statistical signal. And so this is just a flash up. I won't go into all of the details, but Julie is now trying to build a multifactorial model, just like I told you about in the first part, to try to predict what is it about the deleterious genes that makes them deleterious. Some of you may be familiar with receiver operator curves. The bottom line is she can have a fancy model, but really it doesn't really do much better than just gene length as a predictor. So we're trying to understand what that means in terms of the biology and stress. I started this whole talk off talking about stress, and we want to understand stress responses. We think that duplication of some proteins is stressful for cells. We still don't really understand why. Okay, so now I just have 2 more slides. But I think this is a good thing to end on for an evolution seminar. So we are starting to understand a little bit about the fitness costs of duplicating chromosomes, at least in this strain background. And you'll remember I started off talking about why that's important for evolution. If there is if the benefit of duplicating a chromosome in a given environment outweighs the cost of duplicating the chromosome in that environment, that means overall that state of aneuploidy is going to be beneficial. This was the summary of what I talked about before. I'm just going to move on to get to this point. Imagine, though, everything we've done so far is in one strain background. And I just said for evolution, if cells can overcome the cost and there's a benefit, then that individual is going to be more fit than the others in the environment. But imagine, what if we just happen to pick a strain that's super resistant to aneuploidy? The cost is maybe if we look at another strain that's much more sensitive to chromosome duplication. For example, the lab strain I told you about, really sensitive to chromosome duplication. You would have to have, that strain would have to have an enormous benefit to duplicating a chromosome to overcome that cost. So what it means is that if different strains vary in the cost side of the aneuploidy, it means that those different genetic backgrounds may have differential access to different evolutionary routes. Maybe they can never evolve through duplicating chromosomes because they just can't overcome the cost. Those individuals would have to evolve in a different way. So we now are very interested in, does aneuploidy tolerance vary across different genetic backgrounds? And I'll just briefly highlight, and this might have been the paper, I can't remember what paper I picked for us to discuss afterwards, but it might have been Eduardo's paper. So this paper by Scopel et al., done by Eduardo, who's sitting in the audience, when Eduardo was in a different lab. So this is one of the great things about academia. We collaborated with Eduardo and our collaborator, Duda Bensassen, and then later Eduardo got his PhD and came to our lab, which was great. But in this study, what we looked at was, we actually just analyzed another lab's data. So this paper by Peter et al., is this showing up on this diagram? They sequenced 1,000 genomes of 1,000 different Saccharomyces cerevisiae strains. And these strains come from all across the globe. Some are wild, some are used in industry. And they had annotated, and so this is a phylogeny if you can't see it very well, but they annotated which of these strains on the phylogeny has duplication of one or more chromosomes. That's what the little black bars mean. And so part of what Eduardo beautifully did in this bioinformatics study is showed that certain clades of yeast, so group populations of Cerevisiae, they're all the same species, but we have genetic populations. Some of those populations have much more aneuploidy than others. And so I'll skip over that. And so that, what does that mean? So one of the things we are very interested in is, maybe it means that some genetic backgrounds are more tolerant of chromosome duplication, and therefore you see in nature that those strains from that lineage are more likely to be aneuploid for different chromosomes. It's not like they've all fixed one chromosome duplication. These lineages are carrying a bunch of strains with a bunch of different, the individual strains have amplified different chromosomes, okay? So we now have a grant that we're working on for all of the work that James and Julie did that I talked about in the main part of my paper. We would now like to do exactly the same thing, but in a bunch of different genetic backgrounds of strains picked from across this phylogeny. So with that, I think I will end on the slide that I started with, which is the acknowledgement for all of the people in our lab. A subset of is shown here. And I think this has been really a super fun series of projects in the lab, because our lab relies on input from everybody, even people not studying aneuploidy, to really formulate these ideas. So extra super thanks to people in our lab, several of whom are in the audience. And otherwise, thank you to you guys. I'm happy to answer questions. I think we have some time for questions. Nathaniel can't even wait. His hand is like shooting up. I'll do Nathaniel first. And
There may be factors that might matter for how the bacteria to try some of these are. I missed if you saw anything to the origin replication. No, and what about not the number of origins, but the average replication timing?
do you have replication timing in? Well, I don't know if there is a replication time per origin. Sorry, the number of origins with either early or late firing. Is that what you mean?
I mean, I don't know how much they differ in just their average replication timing. Presumably, there's some variation there. Like that, on average, some chromosomes will be the last to replicate. Anyway,
I don't know if that's true on a chromosome level. It may be, but for sure that
exactly synchronized because depending on how many origins there are and how soon they all fire.
Yeah, I'm not sure we looked at it in that way, but the number of origins wasn't chosen. Yeah.
Number of origins is probably pretty tightly correlated with chromosome length, I guess.
So I normalize every user features to the genome. So there's a way to do this is correlated, and some things that is not good for any type of family. You can't take a feature that we are all correlated. So everything is normalized, and that will be one correlation between the features.
And then there must be some features that are so idiosyncratic to a chromosome that you can't really include them in your model. I noticed chromosome 12 was an outlier to the wild type, and that's got the RDNA array. And so you don't have other chromosomes with that array, and that might make it hard to determine whether that's an issue.
Yeah, so that's an excellent, Nathaniel is raising a really important point. We are trying to use the model to make the predicted fitnesses match the measured fitnesses. So you might have noticed, like Nathaniel did, the strain with the chromosome 12 duplication, the point is always above the line, meaning that aneuploid is doing better than the model predicts. And it happens to have a single giant cassette of rDNA. That is DNA that encodes the rRNA, that's RNA that's part of the ribosome. So there is only one chromosome that has the rDNA locus. So if you put that into the model's going to say, oh, this is great. Chromosomes with the rDNA locus do better than expected, and therefore rDNA is important. But there's only one point. There's only one point that that is true for. So this is a good warning about why it's important to know how modeling works, because you can very easily overfit your model in a way that is just has nothing to do with biology, just has to do with the numbers. Yeah, but it did not escape us that the rDNA locus is on chromosome 12. We've talked about trying to test that. James has actually tried several times to delete just the rDNA locus, and it's hard to do because it's a big repetitive region.
And so we'll have the question whether the total number of repeats in there, how that relates to new growth. My last one, just quickly, was gene essentiality. No,
I don't. In fact, I think those are more likely to be neutral, right, Julie?
Yeah, I just whether genes control
So here I skipped over some of this at the end. Where do I have that? So the other thing that sorry, this I should really highlight because I made a big deal about the balance hypothesis. The hypothesis is that genes that encode proteins that are part of complexes are going to be more deleterious because they screw up the stoichiometry, the balanced abundance of those proteins is important. So everybody has talked about the balance hypothesis, but there's essentially no signature. Again, if we look at which genes are deleterious when duplicated and compare them to the neutral genes, the neutral when duplicated, there is no signature for genes that are part of complexes. And now multiple studies have shown that. It's important to think about why, though. And actually what comes from the literature now in recent studies, it probably is really bad for there to be imbalance in certain sets of proteins. So the ribosome, we actually already know if you force the cell to make way too much of specific ribosomal proteins so that there's imbalance, it's actually very bad. But guess what? The cell has figured that out already. So for genes where the stoichiometry, the abundance is really, really important, cells have ways to buffer that. So if you duplicate the gene, what people are starting to realize, if you do proteomics, you don't see that that protein is any higher than when you don't duplicate the gene. Why? Because the cell is very tightly controlling the abundance of the protein. So the balance, so does that mean the balance hypothesis is correct or it isn't correct? It depends on what level of question you're asking. Is it deleterious to duplicate the genes that encode multi-subunit proteins? Probably no. Is it important for if there's imbalance in the encoded proteins? Yes, probably because of the balance hypothesis, right? So it depends on how you frame the question, whether the answer is it correct or not. To the cell probably really does care about the abundances of specific proteins. Yes. Actually, we'll go with you first and then Carol. Yeah.
Yeah,
You in the, yeah.
Okay. Yeah, so I am interested in why tRNA duplication might be beneficial for the cell, and I'm thinking perhaps tRNA, increased abundance of rare tRNA can essentially erode the effect of selection on codon usage bias, and perhaps that's gonna overall, so that explains kind of the mixed effect of the chromosome. Maybe certain chromosomes have tRNAs that are more already abundant, and then having more of them might be more deleterious, but then for certain chromosomes it might have the rare ones, and then if those increases, that might essentially let the cell more be more the mutation the negative selection on the mutation will be negligible for these cells because they have more of the rare tRNAs, and maybe that increases the amount of space where the mutation could.
Yeah. That's a great question and great ideas. So in fact, we were very focused on several of the things that you mentioned. And so some tRNAs, there are a bunch of copies of the same tRNA that recognize the same codon. Those are like abundant tRNAs. And often those tRNAs recognize codons that are very frequently used in the whole transcriptome. Then meanwhile, you have other codons that are very rare. And then their corresponding tRNAs are also, maybe there's only one gene in the genome that encodes that tRNA. So you have rare tRNAs and rare codons. And so your question has to do with maybe actually it's the rare codons that are beneficial to only chromosomes that don't have those rare codons or whatever. And I think Julie, we tried looking at that in a bunch of different ways. She tried in modeling, splitting the tRNAs up into rare versus abundant, and then no signature there. We tried in the latter part where I told you we're trying to use machine learning to predict why are some genes deleterious. We looked a lot at codon bias or amino acid composition of the proteins, and really not finding anything there, which is interesting for other evolutionary theory reasons. We thought we would see more there. So then maybe I can just wrap this up by saying, why might tRNAs be beneficial? The first thing is you're exactly right. Maybe not all of the tRNAs, certain tRNAs are going to be beneficial. You could imagine that if you duplicate a chromosome that's got a whole bunch of one particular rare codon, producing more of the tRNA that recognize that codon could be good, right? There's limitation in the abundance of rare tRNAs. So you could see that that might be a limitation, but we have not seen that signature. What's interesting to us, or to me anyway, is that it's actually, we see a signature for the SSD minus aneuploids. And so you'll remember, I didn't go too much into detail about SSD1. It's an RNA binding protein. Several studies have suggested that it's important for efficient translation. And in fact, there's what's called a genetic interaction. If you have cells that are missing SSD1, they're super sensitive if you make a second mutation in a type of complex that can chemically modify tRNAs. And that chemical modification of this specific set of tRNAs helps the tRNA bind, the anticodon and codon bind really well. If you can't do this particular tRNA modification, the tRNA doesn't bind as well and you get errors in translation. And SSD1 is super important. If you can't make that tRNA modification and you don't have SSD1, cells are like really, really, really sick. So what that means is it implicates that SSD1 maybe has something to do with tRNA or translation. So I'm very interested in that and this connection here. But so it points us to maybe translation of the extra proteins as being maybe an important factor in the why part that we're still trying to figure out.
I sense there's probably maybe a few more questions in here, but I'm gonna have to cut it off here. So please give
Carol, coffee. Oh, okay, good. Great. Well, thank you guys. And do we stay in here for the class? Perfect. Oh, shoot, they were.